Hello everyone. So now let's come to the next topic, that is amelogenesis. Now the word genesis means the formation. So the formation of ameloblast is called amelogenesis. As simple as that. In our previous section, we have discussed about the life cycle of ameloblast. That was pre-secretory stage, secretory stage and the post-secretory stage. So same thing, it's interconnected here, that is amelogenesis. So what is amelogenesis? Going by the definition, it is the process of formation of enamel, and this amelogenesis involves two steps. One is matrix deposition and second one is mineralization. This you can just compare to a mason who is making the buildings. He will bring the cement, stone, sand, everything he will mix. So when he is making a paste of the cement, that is matrix deposition, and he will label it and he will make sure that no one steps over that. A little bit of watering will be done for the cement to become hard. So that process here is called mineralization. So matrix deposition, step number 1, and mineralization is step number 2. So now let's come to the first step, that is matrix deposition, enamel matrix deposition. So as in our life cycle of ameloblast, we have discussed the secretory ameloblast. So the same secretory ameloblast, they start secretory function only after a layer of dentin is deposited. That is their reciprocal induction. They have mutual understanding. Enamel will be formed only if a layer of dentin is formed. So that is very much compulsory. So enamel formation begins in the cusp tips. So when the enamel is going to form, it is in the cusp tips. So before that, the secretory granules. So now we have got a secretory ameloblast and then we have got all the granules. So these granules are packed with enamel proteins. Your amelogenin, non-amelogenin, all these are enamel proteins. So the secretory granules are packed with all of them, rich in proteins, and they migrate towards the basal plasma membrane and release the matrix protein against the newly formed dentin. So they just spit it out, just like exocytosis. Exo means outside. So the cells will just vomit the granules which are rich in these proteins, and that process is called exocytosis. So by means of exocytosis, the secretory granules liberate out enamel protein. So the matrix is being formed over there, and once it is formed, then the enamel formation starts or begins in the cusp tips. We have tooth, we have got cusp. So in the cusp tips and incisional edges, from which it progresses from outwards and cervically. A crown is there from outwards and it comes towards the cervical area. As the matrix deposition progresses, the ameloblast move outward away from the matrix. So they are deposited there, and then once the matrix is deposited, ameloblasts slowly move out of it. So that is what you need to know in enamel matrix deposition. Now in the early stage of amelogenesis, the enamel matrix consists of 20 to 30% of proteins. Initial stages of amelogenesis, it's highly rich in protein, 20 to 30%. But at this protein, which is 20 to 30%, gradually decreases during the process of mineralization. So at the time of matrix deposition, the protein is 20 to 30%, and when the mineralization process is going to begin, the protein number decreases. So that is what you should be knowing it. And this ameloblast, they synthesize and secrete the enamel matrix. So the enamel matrix has a composition. So the composition is what? It is nothing but enamel proteins. And these enamel proteins are two types: amelogenin, non-amelogenins. Amelogenin is the major contributing factor which accounts up to 90%, and non-amelogenin is 10 to 20%. So these two things you should be knowing. Amelogenin and non-amelogenin, both are proteins. So the examples of non-amelogenin proteins are enamelin, tuflin, ameloblastin, and some enzymes like proteinases. So they all have equal role. First and foremost, the amelogenin, which accounts to 90%, they are characterized by presence of large amount of proline. Amino acids proline is very much on them. Whereas the enamelin, what we see has non-amelogenin, has less amount of proline when compared to amelogenin, but has greater quantities of glycine. So your enamelin has greater quantity of glycine than proline, whereas amelogenin more of proline. And these amelogenins are located in the intercrystalline spaces and are involved in the control of crystal growth. So that is their function. Now coming to the secretory phase, which we have already discussed in the life cycle of ameloblast. So in the initial stage of secretory phase, the ameloblast have a flat basal region. Flat basal region. This is the area, flat basal region. After a little thickness of enamel matrix is deposited, this ameloblast develop a conical process at the base. So this is that conical extension what is being formed here, and this conical extension is called Toombs process. Toombs process. So this is a very important structure which is being formed here. The conical elevation from the ameloblast which has been formed is Toombs process. So this Toombs process, once it is formed, lot of activities happen. So in the secretory ameloblast, after the formation of Toombs process, now this cell will have dual function, a dual secretion. The Toombs process contains some secretory granules, microtubules, microfilaments and mitochondria. Once the Toombs process is formed, it extends behind, it comes behind the distal junctional complex and is the only cellular site to interact with the growing enamel surface. Matrix deposition and mineral transport takes place only through this. So what we are going to learn here is, once the Toombs process is formed, so from here only matrix deposition is going to take place and mineral transport is going to take place. So this is a very, very important landmark event happening at this stage, Toombs process. And once Toombs process is formed, dual function it has got. So from this site only matrix deposition and mineralization is going to be initiated. Now this enamel secretion. So once the Toombs process is developed, the enamel secretion takes place through two sites in the Toombs process. So there are two gates. From two places this enamel secretion will form. So the first one is at the interrod growth. First one is interrod growth region formed by proximal end of Toombs process of ameloblast. So the first one is interrod growth region formed by proximal end of Toombs process, and this site where it is located adjacent to distal terminal bars. This is your distal terminal bars. So from the distal terminal bars in and around, the site is located adjacent to distal terminal bars all around the cell, and thus results in accumulation of enamel matrix between adjacent Toombs process to produce interrod enamel. So next to the distal terminal bar, this is the place where your interrod enamel is being formed over here. This is site number 1. Next, coming to site number 2, the other site is the rod growth region located at distal portions of Toombs process of each individual ameloblast and is associated with formation of enamel rod. So this is the second place. This is the place number 1. This is place number 2. So from here also, this area also, that deposition is going to take place. So at the border between the rod and interrod enamel, a sheet-like zone with slightly more concentrated organic matrix persists and demarcates the rod and interrod region. So if you can see this diagram, so interrod and enamel rod, these two are there. Between them, a space is created. So that is what it is to be noted here. Thus, Toombs process is responsible for the rod structure of enamel. What is rod? Our enamel is made up of millions of rods. The basic structural or functional unit, you can call it. So Toombs process is responsible for the rod structure of enamel, and rod structure will be in the enamel. So enamel will be formed only if Toombs process is there. So deposition from the proximal end precedes the deposition from the distal surface. You have site number 1, site number 2. This is site number 1, this is site number 1, this is site number 2. Two sites are there. So the site number 1 precedes the deposition from site number 2. Therefore, the rod enamel is deposited into a pit created by the adjacent inter-rod enamel. So basically, Toombs process is there, and you have got next to the distal terminal bar a site is there from where inter-rod enamel is deposited. And second one, from the distal surface, you have got the enamel rod being liberated out. So till here, we were discussing about maturation of matrix. Now is the mineralization of enamel matrix. Mineralization, maturation over, deposition over. Now it has to become hard. So if it has to become hard or mineralized, then inorganic constituent should come into play. Calcium should come into play. So mineralization takes place in two steps here. One is immediate partial mineralization, and second one is once it is mineralized, it has to mature itself. So mineralization of enamel occurs extracellularly by deposition of minerals. So outside the cell also, lot of minerals should come and they should aid in that. That is secreted by protein matrix. So extracellularly, minerals will be coming from the protein matrix. And during enamel formation, secretory ameloblast is directly involved in both the production of enamel matrix and mineralization. So your secretory ameloblast does the dual function. They will deposit the enamel matrix also and mineralization also. So now we came to know there are two players. First is secretory ameloblast who will aid in mineralization. Second one is extracellularly minerals will come. And for all these things, calcium is required. So calcium is required for mineralization through the circulation to the tissue fluids. This calcium which comes from the tissue fluid is actively transported into the cell. So it is just a VIP. It has, once the calcium has come, it is actively transported into the cell where it gets binded with the calcium binding proteins. So this is the basic steps what you should know. Mineralization takes place, one is extracellularly around the cells, two from the secretory ameloblast itself does a little bit of mineralization, and calcium is required for this. Once the calcium comes from tissue fluids, it is actively transported into the cell. So now the complex of calcium is there and calcium binding proteins. They move towards the distal cytoplasm. So they came inside, the calcium came into the cytoplasm and they move towards the distal side of the cell, from where it is released, and then the free calcium ions are extruded to the enamel matrix by calcium ATPase enzyme. So from the distal portion, this calcium is released with the help of an enzyme, that is calcium ATPase in the plasma membranes of Toombs process. So that is how the mineralization starts up. So now once the mineralization has started up, now the mineral deposition in the enamel matrix will occur in 4 stages. Step by step it will happen. So the 4 stages are primary mineralization stage, secondary mineralization stage, tertiary mineralization stage, and the 4th mineralization stage. So each step is of equal importance. So coming to the first stage, the primary mineralization. So here what happens, primary mineralization, it is an immediate mineralization. The moment matrix is laid down, 30% gets mineralized instantly. So primary mineralization, it is the immediate partial mineralization. Up to 30% of the mineral deposition, immediately mineralization happens. That is primary mineralization. Coming to the second one, that is a secondary mineralization. This starts at the surface and proceeds towards the dento-enamel junction. So from the surface of the crown, it proceeds and it goes towards the dento-enamel junction. In the tertiary stage, the mineralization rebounds from the inner layer to enamel outwards. It is just the opposite of secondary stage. Secondary stage from outside to inside. Tertiary stage, from inside to outside, mineralization takes place. And in the fourth stage, that is the end stage of mineralization, further deposition of minerals will take place and make it hyper mineralized. More and more minerals will come and already mineralized one will become hyper mineralized. So step number 1, primary mineralization, immediate mineralization 30% will happen. Secondary mineralization, from the surface to DEJ, mineralization will go inside. Tertiary stage, from inside to outside towards the surface of enamel, mineralization will take place. And in fourth stage, more and more minerals will come and make the site hyper mineralization. So these are the steps in or stages in mineralization. Now coming next to the last stage, that is maturation. So mineralization is over. Now it has to mature. The enamel has to mature. It has to become more thick. So maturation is characterized by gradual completion of mineralization. So once the mineralization is over, then gradually the extension of mineralization will lead to maturation. So during maturation, what will happen is reabsorption of matrix protein and water takes place. Water and proteins will be taken up. Initially, the amelogenin proteins are absorbed into specific crystal phases, thus controlling growth. So amelogenin proteins are taken up and they will control the growth. The amelogenin proteins are removed from the mineralizing front by proteolytic cleavage. So once the work is over, they will be removed. So how it will be removed? With the help of an enzyme, that is proteinases enzyme. And after some time, it is again reabsorbed by endocytic action of ameloblast. And in the tertiary stage of mineralization, the maturation process starts at the dentoenamel junction and progress toward the surface. Similarly, it proceeds from the cusp or incisional cusp to cervical region. So, in the tertiary stage, the mineralization will start from DEJ and progresses towards the surface, inside out. And similarly, it proceeds from the cusp or incisional cusp tips to cervical region, that is upside down. So these are the two steps in your amelogenesis: maturation, matrix deposition, maturation, and the mineralization. Now in our next session we will study the structures of enamel. Thank you.